Welcome to Pharma Intelligence's webinar on gene therapies, a new paradigm in curative medicine, the state of gene therapy research and development. My name is Alex Shimmings, and I'm the Managing Editor for Scrip. Today, I'm joined by Natasha Bowleiter and Amanda Miklas. Amanda is a drug analyst with Pharma Projects. Natasha is a drug analyst with Pharma Projects, and Amanda is a principal analyst with Data Monitor Healthcare. Today's webinar will present data from Pharma Project's latest analysis entitled Gene Therapies in a Bottle, Pharma Just Needs to Rub the Right Way, and Data Monitor Healthcare's recent analysis called Trends in Gene Therapy. Please note that owing to the slightly different publication dates of each analysis, there are different snapshots of data represented in the figures included. Looking at today's agenda, first we'll be reviewing trends in gene therapy development with a look at how the pipeline has changed over the past few decades. Next, we'll review the current state of the gene therapy market, looking at who the key players are and which companies have succeeded in getting a gene therapy product to market. Then we will touch a little on pricing and reimbursement, a topic that will be discussed in more detail in a future Informa webinar. After that, we will move on to assess the clinical development landscape, looking at the most active therapy areas, including a breakdown of the pipeline by the different approaches being taken and delivery vehicles used. And then we will take a deep dive into both oncology and non-oncology gene therapies. Finally, once we have concluded, we will have time for a brief period of questions and answers. If you wish to ask any questions during the webinar, please enter them in the box below. For any questions that we don't address during today's session, we will be sure to follow up via email. So let's get started. Natasha, can you begin by telling us how the volume of gene therapy candidates in development has changed in the last 20 years or so?
Yeah, sure, Alex. So, gene therapy is making something of a comeback and being heralded as the next big thing in drug development for the second time around. Taking a look at the graph, we can see that interest in the area first began back in the 1990s and quickly gained momentum as shown by the upward trend in the total number of gene therapies in active development during this time. But following much publicised clinical trial fatalities, it was widely believed that investment in the area had totally dried up. And as the graph shows, there was a slight dip, but research in this area never really went away. In the last 2 years alone, the total number of gene therapy projects in the pipeline has doubled. This slide shows that there is a total of 6 gene therapies available on the market today. Arguably, the most notable of these products is Glybera, which attracted a lot of media attention when it was launched last year, as it was the first drug of this type to reach the market in the EU. However, it was not the first gene therapy launched in the world. Just over 10 years ago, Genedicine was launched in 2004 in China for the treatment of head and neck cancer. Another cancer-treating gene therapy, Oncoine, followed not long after in 2005 and was also launched on the Chinese market. Next was Rexin-G, launched in the Philippines, targeting solid cancer. And most recently, Imglygic, launched in the US by Amgen for melanoma treatment. The table shows the comparison in the disease focus of the successfully launched gene therapy products and shows the split between cancer-treating therapies versus the rarer, more obscure diseases, such as lipoprotein lipase deficiency, which Glybera treats.
Thanks, Natasha. It's interesting that the pioneering gene therapies were developed outside pharma's traditional markets, isn't it? But I suppose with Glybera being the first gene therapy approved in the Western world, this makes for an interesting case study for the inevitable pricing and reimbursement questions. Amanda, can you touch, please, on some of the market access issues with gene therapies?
Sure, thanks, Alex. We wanted to highlight here a couple of pricing considerations in gene therapy. As Natasha mentioned, Glybera, the first gene therapy for a monogenic disease approved in the Western world in 2012 and indicated for lipoprotein lipase deficiency, has had a challenging time on the market because of the high price tag at just over $1 million per treatment. The high upfront cost for gene therapy is a major challenge from the payer point of view, and Glibera's example highlights this. It's estimated that only 150 to 200 patients in Europe, which is where the drug is approved, are eligible patients. Therefore, the overall impact on the budgets is quite small. But access to patients has been difficult following negative reviews, for example, by Health Technology Assessment, or HTAs, in France and Germany. One potential way to price gene therapies to improve and ensure access as an alternative to an upfront payment is where a pay-for-performance or pay-for-value model could come into play. This mechanism would allow for gene therapies to be reimbursed based on real-world evidence against specific measurable outcomes. One example is GlaxoSmithKline's Strimzelis, which was just approved in May by the European Commission for severe combined immunodeficiency due to adenosine deaminase deficiency. GSK is experimenting with a money-back guarantee in Italy, where the drug will initially be administered for treatment. Strimzelis will cost $665,000, and GSK will get paid in installments, but only if the gene therapy works. Otherwise, it will provide a refund. For payers, another challenge is the uncertainty over long-term efficacy of gene therapy. So there's a concern from payers on paying for that kind of long-term therapy over a patient's lifetime. A potential pricing mechanism that could address this is an annuity. For gene therapies that treat monogenic diseases and are curative, that is, they are a one-time treatment, an annuity type of payment could be used as opposed to a high upfront cost that impacts the payer budget all at once. This is suggested in a study by Troy and Brennan and James Wilson. Payments would be made over time for a certain number of years based on the evidence that the gene therapy is working. This particular study proposed that the original annuity payment could be set with reopener clauses if, for example, the patent expired or a new competitor entered the market that would change the payment, similar to the way that market events would affect a standard drug. A further challenge to payers in this instance, however, is the risk that insurers would take on when patients change insurance coverage and the impact on the responsibility of such a payment mechanism.
Thanks, Amanda. As previously mentioned, pricing and reimbursement of gene therapies will be covered in greater detail in a more commercially focused webinar coming from Informa in the next few months. So with that background, Natasha, can you now give us an overview of the gene therapy pipeline?
Yeah, sure. So this graphic represents the overall pipeline for gene therapy candidates, which are in preclinical and clinical development, all candidates which have had their development halted completely. The active development statuses are shown in purple and include the stages of preclinical through to launched. Meanwhile, the inactive statuses are shown in a lighter pink colour and represent suspended or discontinued projects, or projects which have had no further information released to suggest they are still ongoing. The horizontal bars are showing the total number of candidates at each of the different stages, while the pie chart shows the percentage of the active versus the inactive statuses. At the moment, there is a total of 501 candidates in active development, with the majority in preclinical development, a smaller number in phase 1 than phase 2, and a smaller number still at phase 3. This overall trend is typical of many other drug development research areas, as a large majority of candidates don't progress further than the preclinical stage. Another general trend is seen in the higher number of compounds at phase 2 than phase 1. Although many more compounds reach phase 1 than phase 2, and although there is a high level of attrition between the two phases, there is also usually a build-up in the total number at phase 2. This is because the overall length of the phase 2 stage is longer than phase 1, which sometimes results in compounds getting stuck at this level. In the case of gene therapy, 33% of projects are still at active stages of development, while 66% are no longer active. This is a slightly higher percentage of candidates in active development compared to the pipeline as a whole for all research focuses, which shows approximately 25% of projects at active development phases. This higher than average percentage of active projects is generally quite typical of a newer and less mature area of research.
Thanks, Natasha, for that overview. Now, can we take a look at the different disease groups represented within it, please?
Yeah. So, this figure shows how gene therapy projects have targeted almost every type of disease area, and also shows that cancer and neurological are the two disease groups that research is being pursued in the most. In pharma projects, we also capture data for so-called rare diseases, which can be seen as the second largest bar on the chart. However, it should be noted that indications captured in this category are not mutually exclusive to the other disease groups. In fact, every drug counted in the rare disease bar will also be counted in one of the actual disease groups as well. The focus on rare disease and cancer is an interesting pattern, as the industry is targeting not only the smallest and most niche disease area, but also the largest and perhaps the most important area for the pharmaceutical industry. A number of the gene therapies in development for rare diseases have also been granted orphan drug designations. If a compound manages to gain approval in an orphan designated indication, the company can receive certain benefits. For example, a longer exclusivity period on patents and tax credits.
Thanks, Natasha, for that very comprehensive pipeline overview. Next, let's get into some more specifics on the different gene therapy approaches and delivery vehicles being investigated. Amanda, can you start us off by talking about the different approaches being taken to developing gene therapies, please?
Sure, thanks, Alex. Gene therapy involves introducing genetic material into a cell to correct or inactivate a mutated gene that causes a disease. To enable gene delivery, the two main approaches or categories for gene therapy are in vivo or ex vivo. In vivo involves modifications to genes directly in cells that are inside of the body. Alternatively, an ex vivo approach, which is often referred to as cell and gene therapy, is used when the gene modifications are done to cells, for example, bone marrow cells that have been extracted from the body. Following gene transfer to the cells in the lab, the cells are then reintroduced back into the body. Both in vivo and ex vivo gene therapies may be delivered by various vectors, the development of which has been challenging in terms of ensuring enough of the gene is delivered in an efficient manner and also managing the potential immune responses and toxicity. Vectors for gene delivery may be broken down into viral and non-viral categories, and a list of the common ones being used are shown in this table, alongside whether they can be used ex vivo or in vivo. Viral vectors, or recombinant viruses, bind and introduce the gene to the host cell via the replication cycle. Retroviruses were the first used in clinical trials for ex vivo gene therapy. You can see that the current gene therapies on the worldwide market, including those approved in the US and EU, as well as those approved outside of those major markets, span various types of viral vectors. Rexin G, an anti-cancer gene therapy, and Strimvelis, which was recently approved, are both delivered by retroviruses. Gendecine and Oncorine, also oncology therapies, use adenoviruses, while Glybra is delivered via an adeno-associated virus. And Imlygic, which is a genetically modified oncolytic virus therapy, is an oncolytic herpes simplex 1, the simplex virus 1 derivative. The only marketed gene therapy currently delivered via a non-viral vector is Neovascogen, a drug sold in Russia by Human Stem Cells Institute. Non-viral vectors include, for example, naked DNA or DNA complexes used for gene delivery. These may be advantageous over viral vectors in that they may be produced at a larger scale and have low host immunogenicity.
Thanks for that overview, Amanda. It would be great now if we could take a look at how the pipeline breaks down in terms of these approaches and delivery methods, both by phase and by therapy area, please.
Sure, thanks, Alex. So in terms of breaking down the gene therapy pipeline by these approaches and delivery methods, you can see here in this chart that they vary across diseases, which are listed on the x-axis horizontally, with the pipeline volume plotted on the y-axis. The columns themselves are split up by in vivo or ex vivo approach and delivery vehicle. As Natasha showed earlier, oncology is the busiest area for gene therapy drug development. The majority, approximately 89% of ex vivo gene therapies in development are for cancer. Most of these oncology candidates are in preclinical studies, and many are engineered autologous or allogeneic T cells. In all other diseases, including monogenic, neurological, infectious, ocular, and cardiovascular conditions, viral vector in vivo gene therapies are being tested. Focusing in now on just the in vivo gene therapies in development, this shows a breakdown of diseases by the viral vector used for delivery. While oncology leads in the total gene therapy pipeline by volume of drugs, in this subset of just in vivo gene therapies in development, most are being tested for monogenic diseases, then followed by cancer. You can see here that the vector used also varies among the diseases. The different vectors are represented by the color split in each of the columns. The adeno-associated virus is the vector of choice for gene therapies in most diseases, including monogenic diseases as well as neurological and ocular disorders. The in vivo gene therapies in development for cancer, on the other hand, are using adenoviral or other types of viral vectors. Adenoviruses may be more effective for cancer because of their ability to elicit stronger immune responses against tumor cells. This is a side-by-side comparison of in vivo gene therapies in development, comparing just the group of preclinical candidates, which are shown on in the right-hand figure, with those in phase 3 or later, shown in the left-hand figure, in order to highlight some of the differences between the two. Most advanced gene therapies are in development in cancer and use adenoviral vectors, while in the preclinical pipeline, most of the candidates are focused on monogenic diseases delivered by adeno-associated viral vectors. Also, notice the shift in cancer development. While most advanced in vivo gene therapies for cancer are using adenoviruses, early-stage cancer drugs are using other types of viral vectors, and in terms of volume, aren't as abundant as the result of the increase in ex vivo gene therapies. In looking at the pipeline of in vivo gene therapies delivered by plasmid vectors, you can see most of the development is concentrated on gene therapy and cancer gene therapies, many of which are either in phase 1 or phase 2 trials. The next most active therapy area for non-viral gene drugs is cardiovascular disease. Non-viral gene therapies in other areas, including ocular, infectious, and neurological diseases, are still in early stages.
So based on the data, it looks like oncology gene therapy development differs in many ways from gene therapies in the pipeline for other diseases. Amanda, would you please walk us through the gene therapies that are in development just for cancer?
Sure. As you mentioned, Alex, the next few slides will focus in on specifically anti-cancer gene therapy and then break out immunotherapy and non-immunotherapy approaches. The two figures shown here outline the oncology gene therapy pipeline by phase, which is shown in the left-hand figure, and by delivery approach, which is in the right-hand figure. As we previously saw, oncology is a lead therapy area for gene therapy drug development overall. The majority of oncology gene drug development is early stage, and the investment in this early stage research is evident in the fact that the universe of preclinical anti-cancer gene therapies has grown fourfold since 2012. In looking at the breakdown of the delivery vehicles for anti-cancer gene therapies, most are being delivered ex vivo via autologous cells, followed by allogeneic cells. For in vivo delivery, adenoviral and plasmid vectors are among the most used by volume. Cancer immunotherapy, which is one of the most heavily invested areas in drug development today, is also being employed in gene therapy. This figure shows various immunotherapy approaches taken with gene therapy. The types of therapies are listed on the x-axis horizontally, with the number of drugs plotted on the y-axis. For each immunotherapy approach, the pipeline volume by phase is displayed in the split columns by color. In looking at the various immunotherapy approaches, most gene therapy candidates in the pipeline that are immunotherapies are being delivered via modified T cells, followed by tumor-associated antigen vaccines and whole cell vaccines. Most of the T cell therapies are in preclinical studies. T cells may be modified with genes encoding certain T cell receptors to help fight off cancer in a number of ways, including resisting immunosuppression and enabling engineered T cells to enter into tumors. In terms of looking at non-immunotherapy approaches to treating cancer with gene therapies, these involve using genes for targeted destruction of tumors without causing severe side effects. This figure, similar to the previous one, shows the options for non-immunotherapy approaches employed by gene therapy on the horizontal x-axis, with a volume charted on the y-axis, and the phase represented in the split columns by color. Most of the development has been concentrated on other types of cytotoxic therapies, as well as oncolytic virotherapy and tumor suppressor therapy. 2 oncolytic virotherapies are already on the market, including Amgen's Imlygic and Shenzhen Cybiano GeneTech's Gendicine, which is approved in China.
Thanks for that overview of both the cancer and non-cancer gene therapies. It certainly is an active area. Natasha, can you tell us now about the most active companies developing anti-cancer gene therapies, and also those companies that are most active outside cancer, please?
Okay, sure. So to begin, I would just like to point out that on this graph, phase 2 is shown in pink, while phase 3 is shown in grey. Overall, this figure shows that cancer-targeting gene therapy development is not being carried out by the biggest industrial players. Although cancer is a main area of interest for Big Pharma, many of the larger companies, as of yet, haven't been willing to invest money in this research area. 2 companies which are currently neck and neck in the anti-cancer space are Newlink Genetics and Transgene, both of which have 2 compounds at phase 2 and phase 3 respectively. Kite Pharma is another big contender with 5 cancer-targeting therapies, all of which are at phase 2. Prescient Therapeutics and Advantagene are also right behind, with a single compound in both phase 2 and phase 3. Just as with cancer-focused research, overall it is the smaller, lesser-known companies that are seen to be taking a larger role in the non-cancer and rare disease space. However, the current frontrunner in the non-cancer area is GlaxoSmithKline, which is the only company within this cohort with an approved product. GSK has developed a stem cell gene therapy to treat adenosine deaminase deficiency, which is now approved in the EU, and also has 2 other phase 2 candidates in the pipeline. Spark Therapeutics has the largest number of candidates in development, with 4 in total. It currently has 3 phase 2 and 1 phase 3 projects under development for a mix of rare eye diseases and haemophilia B. Bluebird Bio has the largest number at phase 3, one of which is the widely hyped Lentiglobin for beta thalassemia, which has shown very positive trial results and has the added benefit of allowing multiple months between treatments, in contrast to the standard practice of monthly blood transfusions. Another compound, which has also recently shown promising results in a phase 2/3 trial, is Janssen's candidate for Leber's hereditary optic neuropathy. A company which only has 1 project in the pipeline and is therefore not shown on the graph is Molmed, with a candidate for graft versus host disease, which recently received EU COMP positive opinion on the orphan drug status for adjunctive treatment for haploidentical HSCT, which stands for hematopoietic stem cell transplantation.
So it seems that the gene therapy charge is being led by the smaller biotechs that specialise in this area then. Amanda, can you now talk about gene therapies in development for acquired diseases?
Sure, thanks, Alex. For diseases outside of cancer that are the result of acquired genetic mutations that occur over a person's lifetime, as opposed to inherited genetic disorders, there are gene therapies in development to target several conditions in the infectious, cardiovascular, ophthalmic, and neurological disease areas. Within infectious disease, there is a large concentration of gene therapy candidates being tested for the treatment of HIV, including 5 that are in phase 2 trials, which are shown in this table, alongside their company sponsors, targets, and likelihood of approval metrics when available. While existing retroviral therapy is successful and has reduced mortality, the drug costs are high and side effects and multi-drug resistance limit their use. As a result, gene therapies are in development and use different approaches, including RNA interference and antisense gene sequences to viral targets, as well as targeting 2 chemokine receptors, CXCR5 and CCR4, which are host co-receptors for HIV-1, with the goal of disabling viruses and preventing their cellular uptake. Calimmune, a privately held U.S. company, has been quite active in the HIV area, working on 2 of its own treatments targeting chemokine receptor 5, as well as in-licensing from Benetech Biopharma a candidate against the same target. Sangamo's SB728, which also interacts with chemokine receptor 5, has the highest likelihood of approval among these phase 2 gene therapy candidates at 32%, according to Biomed Tracker, and this is 5% above the average for this phase in this indication. This increase is attributed to strong results showing that SB728 yielded an increase in CD4 count in patients with undetectable viral load but low CD4 counts, further emphasizing that genetic modification could reprogram T cells for HIV resistance. Sangamo expects to report out results at the end of this year from 5 additional patients enrolled in a 3rd cohort of its phase 1/2 trial, and based on that data is looking to possibly partner the drug for future development and commercialization.
Thanks, Amanda. So in terms of other advanced gene therapy candidates that are in late stage development now, Natasha, can you tell us which ones are the lead ones outside oncology and what indications are being pursued there?
Okay, sure. So there's quite a range of non-oncology indications being looked into. Some are rarer diseases, while others are more common conditions, such as arthritis and various cardiovascular indications. A number of the current phase 3 candidates have received fast track and or breakthrough statuses from the FDA, which is promising for this new area as it recognises the potential of gene therapies to meet unmet medical need or to be able to provide substantial improvements over existing therapy options. Most recently, Bluebird Bio's Lentiglobin has received fast track designation for beta thalassemia and sickle cell anemia, as well as recently receiving breakthrough status for beta thalassemia in the US. CardioNovo received US fast track designation for the treatment of myocardial ischemia back in 2007, followed by Calatogene, which received fast track designation for critical limb ischemia in 2010. US fast track also went to Rayon Pharmaceuticals' VM202 candidate for the treatment of myotrophic lateral sclerosis, while the FDA also awarded breakthrough designation to Spark Therapeutics candidate for inherited retinal dystrophies.
So, in conclusion, we've heard today that the gene therapy area is now poised for growth despite the failure of a number of candidates back in the late 1990s and the early noughties. We've also seen how the next wave of approved drugs is likely to target multiple rare diseases. Also, considering the large volume of early-stage oncology candidates in the pipeline, it seems reasonable to suppose that gene therapies for cancer indications will continue to represent the lion's share of the pipeline, especially as research continues into the ex vivo modification of T cells. And finally, GSK and Sanofi are the leaders in this area out of the big pharma firms, but for the most part it is the smaller biotech companies that are leading the charge in this area. So thanks again to Natasha and Amanda for this review of the gene therapy pipeline. Now we'll take a pause for a few seconds to see some of the questions that have come in. So the first question we have is on the CAR-T area. CAR-T therapies are one of the big areas of biotech investment today. Where do CAR-T therapies fit into the gene therapy pipeline? Amanda, I think we'll pose this one to you, please.
Sure, thanks, Alex. So CAR-T therapies are considered part of the modified T-cell group of ex vivo gene therapies in the pipeline, which was the figure shown earlier where we had the anti-cancer gene therapies broken out by immunotherapy and non-immunotherapy approaches. So the CAR-T therapies would fit in with the modified T-cell category in the immunotherapy categorization. These T-cells are modified with genes encoding chimeric antigen receptor, or CAR, which is where the CAR-T acronym comes from. And the majority of genetically modified T-cells in development are CAR-T therapies. Novartis is one of the companies that's most advanced with a CAR-T therapy. It has an autologous CAR-T therapy targeted toward the CD19 protein, and it's in phase 3 trials for acute lymphocytic leukemia and B-cell lymphoma. Novartis actually got this technology from the University of Pennsylvania several years ago. One thing to note is that Novartis recently announced that it was dissolving its cell and gene therapy unit and reintegrating some of that research with its oncology R&D. However, the company has stressed that it is still committed to this CAR-T program targeted towards CD19, and they still plan to file it for approval in the US and EU sometime next year. And another thing to note is that Novartis isn't the only company that's in late stages of the CAR-T therapy. It's in pretty strong competition with 2 biotech companies, Juno Therapeutics and Kite Pharma. Both of them have late-stage CAR-T therapy candidates in their pipelines. And Juno Therapeutics has actually partnered its technology with multiple companies, including Celgene. And Juno was recently given the go-ahead to move forward with its CAR-T therapy called JCAR015 after a temporary clinical hold was lifted by the FDA. CAR-T is definitely a very busy area for development right now.
Yeah, it certainly caused a bit of a stir with script readers when Novartis made its announcement, definitely. CAR-T stories are always very well read. So thanks, Amanda, for that. The next question we've got coming in is, what is the next gene therapy likely to be approved? I think we'll give this one to Natasha.
Okay. So at the moment, there are no gene therapies in the pipeline listed at pre-registration status. So it'd actually be better to look at the compounds currently at phase 3. Perhaps something like lentiglobin, which following its promising trial results could be filed for approval in the near future. Also taking a look at data from our sister product, Biomed Tracker, lentiglobin currently has a 69% likelihood of approval for anemia, which is actually 9% above average for this sort of indication.
So that's looking promising then for them.
Yeah.
Great. Our next question is on gene delivery. You've talked about viral and non-viral vectors, but are there next generation vectors being developed for gene therapy? I think, Amanda, that one for you.
So in the webinar, we talked about the viral and non-viral vector approaches to delivering gene therapy, and these are what are commonly being used now in the pipeline to develop gene drugs. One possibility that's being discussed are bacterial vectors as a next-generation vector, but because these are larger and more complex than viruses, they're not being used in treatment now, and more work needs to be done to improve safety and efficacy for them to be used. But one area that bacterial vectors might have potential in is oncology, because bacteria that's systemically administered can naturally home to tumors and replicate locally at high levels. There's also been some discussion on using adult mesenchymal stem cells as a way to deliver genes for long-lasting production of therapeutic proteins. And these cells have low immunogenicity and immunosuppressive properties that make them options for this type of delivery.
Cool, that's some really interesting science going on there. So our next question is, what is the main reason for gene therapy projects to be discontinued? Interesting. Natasha?
So out of the disclosed reasons that we have captured in farm projects, the main reason for discontinuation is strategic reasons. The second most common reason being given is financial, and efficacy issues are third. So it's quite interesting that the most commonly disclosed reason for discontinuation is strategic, and this may be a reflection of the risky nature of pursuing development in this field.
Interesting. Thanks, Natasha. Right, it looks like we've run out of time for more, but please send us any additional questions you have to pharma@informa.com, and we'll be sure to follow up after the webinar. So once again, I'd like to thank you all for joining this webinar, and thanks to our panelists, Natasha Berleiter and Amanda Miklas. A recording of the webinar will be available shortly, and as a final reminder, Informa Pharma Intelligence will soon be hosting another webinar on the commercial aspects of gene and cell therapies. This webinar will review key players and financing and partnering trends, so please look out for details. Thank you.